my side. I'm Urh and Vienna is there, so we are from Urh Cernigoy, so the producer of chromatographic columns. And we jumped into this project somewhere in the middle. And actually, even the title suggests that actually for us this is a step forward to the production of monolithic columns for diagnostic applications. We are mostly producing and we are focused on preparatory chromatography, so for downstream processing, so big scale of this biotechnology, biotech and pharmaceutical companies. But last years we started somehow even to tip in this field of the small volume columns. And within this, let's say, project, we had kind of 4 different goals. One is to focus on enzyme reactors, so the imers, so immobilization of the enzymes. The second is to immobilize titanium dioxide nanoparticles, so actually to develop monolithic columns with immobilized titanium dioxide. The third one is to produce and develop human serum albumin depletion monolithic column, and the fourth goal was to go with the miniaturization of these columns. So before I didn't mention we are the producers of chromatographic columns, but our columns are innovative in the way that they are monolithic columns. So they are not beaded columns, but the composition of the material is the morphology is different. So it's one piece of material with the convective pores inside, what enables some advantages towards the classical chromatographic columns. So if we go into a little bit deeper detail of each of these goals, the first goal was to develop efficient, stable, and selective immobilized enzyme reactors. From the beginning, there were suggested 2 enzymes, pepsin and trypsin. With the pepsin, actually, we didn't do a lot of work, we just did some preliminary studies, but then we focused more or less to the trypsin columns, the preparation of an optimization of mobilization of trypsin. So for the pepsin, just one SDS page, so here is the sample of hydrogen, here is then after the treatment. We just showed that, we showed that the reactor, actually the enzyme, worked, but I don't think if you even tested sometimes I can't, so you did, okay. But then the most of this work on the imers was really done on the trypsin monoliths, and we did a very thorough study. And we started from the activation chemistry, so we tried different activation chemistry on the monoliths, and then we somehow adopted and developed a method using small substrate, which is used in standard determination of trypsin enzymatic activity, and also other immobilization parameters were thoroughly studied. We worked everything with the aim to obtain really the best trypsin column. So just to summarize some of the results, so the steepness of the curve here shows the activity of the prepared imers. Here are 4 different activation chemistries. This one with the highest activity was obtained on aldehyde-based column, and actually we focused on the work later on this type of activation chemistry. We also were able to show that different reductive elimination procedures, so immobilization conditions, very affected the final activity of the imers. So we did a more thorough study of exactly how much of the trypsin is immobilized on the column in order to get a correlation between the amount of the trypsin immobilized and activity, which is then later obtained. So we developed a method, HPLC-based method to do the mass balance of the trypsin, and we found out that with our classical monoliths with 4 4 sizes of 1.5 micrometer in diameter, we could immobilize maximum around 6 milligrams of trypsin per milliliter, but it's actually a very big amount of density of the ligand if you compare it with what you do in solution, for example. So that's why even the expected activity, not per milligram of the trypsin, but overall activity should be higher than in solution from this point of view. Then we also tried to figure out how if we could obtain higher activity with immobilizing higher amount of trypsin with the way to use the monolith with the lower core diameter, so with higher surface area, but still keeping the monolithic properties. And truly, because the surface area of the monolith with 600 nanometers diameter, it's approximately double of the surface area of the classical one. We also doubled the amount of immobilized trypsin, which was around 1 to 1.2 milligram, around 10 milligrams per milliliter of the monolith. And then because we got all the data, and we used this the measurement of the activity of the immobilized trypsin, and we correlated this with immobilized trypsin, and we obtained a good correlation with this classical monolith. So the amount of the immobilized trypsin correlated with the activity of the IMER, and with the 300 nanometers, because there is more trypsin immobilized, the activity was really higher, but not anymore in this correlation, in the linear correlation. So then, I apologize for the bad design of experiments figures, but I just tried to get from my colleague today in the morning, and we were not able to get a good resolution. But just to have an idea, because we performed then the final optimization of the immobilization of trypsin using the quality by design principle, and we tested, I think, 6 or 7 parameters on the immobilization. We prepared a series of the experimental conditions, and we prepared these columns. We tested them firstly with these small organic molecules, and then they were sent to Medical University of Vienna, and they tested it also with the big protein. And what we were very surprised is that, for example, completely different, so if the red color means the column with the highest activity, with the small molecule, the highest activity is completely with the other conditions than with the protein probe. I think we still have to work on this issue, because I still don't know everything. We can understand something, but I still think that there is something hidden behind what we don't understand. So actually, let's say the optimal trypsin columns were also tested according to the trypsin in solution. So this is the measurement based on the standard measurement of the trypsin in the solution and the activity of BA units per milligram of trypsin. And here is one of the IMERs, not maybe the best one, but at least one of the best what we produced. And you can see that the decrease of the activity per milligram of the trypsin is approximately 3 times slower for the immobilized trypsin compared to the solution, what I still find a very good result. But still, again, I have to say that this is measured with this small organic molecule, and I don't think that this would be still the same with the big proteins which are used in the real proteomics work. Okay, so just the summary of this first part is that we have chosen the aldehyde monolith as the optimal for trypsin. developed these methods, and we did a really thorough study for the evaluation of these imers. And this is, at the moment, this was probably the best what it could be done from our side. Then the second scope was to prepare titanium dioxide columns. Actually, the titanium dioxide with the intention for the enrichment of phosphopeptides, phosphoproteins from the samples. And